So today our focus is going to be on speciation. So what are things that, what the definition of a species is, things that can help create new species, as well as things that help keep species separate and remain as two separate species. So let's start with what a species is. So a species is going to be a population whose members can interbreed and produce viable, fertile offspring. So that's what's going to be key here. They may be able to produce offspring, but if they are not viable, meaning they will survive, and they are not fertile, meaning that they can produce offspring, then that still keeps them as two separate species. So in essence, a species, these things are going to be members of the same species are reproductively compatible. Meaning, again, that they can interbreed and produce viable, fertile offspring. So the example that we see down here in the bottom between the horse and the donkey, a horse, its diploid number is 64, where a donkey's diploid number is 62. So when they produce gametes of 32 and 31. So when a horse and a donkey breed, they result in a mule, which has a diploid number of 63. And so while it produced offspring that was viable, a mule is not fertile. A mule is sterile, which is what keeps a horse and a donkey as two separate species. So for the species to form, for a new species to originate, the populations need to become isolated. And they can become isolated in kind of two basic ways. They can either be geographically isolated, so physically isolated, or they can be reproductively isolated. So even though they may live near one another, like for instance the horse and the donkey, when they reproduce, something is not compatible. Because again, remember, a species is reproductively compatible. And these isolated populations, once they become isolated, they will evolve independently. And since they evolve independently of one another, that can lead to the formation of this new species that would make them reproductively incompatible. So let's talk a little bit about the isolation. So there are two basic ways that the speciation can occur. Speciation is the formation of the new species. So we have allopatric speciation, which is a geographic separation. You can see in the picture here, this river has formed the canyon between the two sets of trees. So these two sets of trees are physically separated from one another. So this is our geographic separation. This is the mountain range that formed and separated them. This is the birds that flew to the island and they don't come back to the other island now. So again, a geographic physical separation. The analogy would be that they are in another country. Where our sympatric isolation or sympatric speciation occurs when they still live in the same area. So I'm in the same country. They still live in the same area. We can see this happening in the trees. But somehow they have become isolated within that area. You can see those trees here that for some reason these lighter green trees are no longer reproductively compatible with those dark green trees. Maybe that's because they were having too much competition. Maybe they changed the way they get pollinated or their seeds are dispersed. For whatever reason, though, they are a whole different species now. They're reproductively isolated, yet again, they're still living in the same location. And we'll look at some examples of this and how this would work. So we're going to look at our focus now is going to shift over to the barriers that help maintain this reproductive isolation. Okay? And so we have barriers are grouped into two basic categories. We're going to have either prezygotic barriers or postzygotic barriers. So a prezygotic barrier would be a barrier that prevents the zygote from ever forming. Remember, the zygote is the sperm in the egg. Right? This is what is formed after fertilization. And so a prezygotic barrier prevents this zygote from ever forming, prevents the sperm and the egg from ever reaching each other, and from fertilization from occurring. So a first example of a prezygotic barrier would be the geographic isolation that we talked about on the previous slide. So the species are occurring in two totally separate areas. Okay, these little fox organisms here are separated by the river. Okay, so they have a physical barrier, and this is going to result in our allopatric speciation, which over time can result in that reproductive isolation and therefore the formation of a new species. These two, this fox and this fox, could have been separated and for generations. This family lives on this side, this group lives on that side, generations and generations and generations, years and years and years and years and years and years and years, there's these two populations of foxes on each side of the river. And if those two ever meet again and they're able to reproduce and have viable fertile offspring, then they did not become a new species. But if they are not, then the speciation occurred. We can also have what's called ecological isolation. So ecological isolation is part of that sympatric speciation. So the species are occurring in the same region, but they occupy different habitats. So they rarely encounter one another. Okay, they have a separate niche. If you remember, a niche is where they fit into the environment, what their role is within the ecosystem. And so the example we have over here are these anole lizards that live in the Caribbean. And all four of these are different examples of these lizards that are in the Caribbean. But they each have a different niche or a different role that they fill in the ecosystem. Like, for instance, this brown one here lives on the trunk of the tree. These live in the grass. These live more out on the leaves. These live out on the branches. And so even though they're living in the same area, they are reproductively isolated, and they have formed their own separate species because they are because they don't encounter one another. Okay? They are active in different areas of the vegetation, and so they don't have to compete with one another. Because if they had to compete with one another, one of them could have possibly eventually become eliminated. Another form of prezygotic barrier is temporal isolation. So this is a time isolation. As a general rule, a breeding time insulation or isolation. So the species are going to breed during different times of the day, either different seasons or different years, depending upon what their cycle is like. Okay? But because they are breeding at these different times, they are not going to breed together. The example in the little cartoon here, right? This one flower here is ready to be pollinated, while this one is not. Therefore, those two are not reproductively compatible, and so they form a separate species. This again would be our sympatric speciation. These organisms can live in the same location, but because they are breeding at different times of the day or seasons or years, they are not breeding with one another. Another prezygotic barrier would be a behavioral isolation. So again, we're looking at these things that are going to prevent the zygote from ever forming. So with behavioral isolation, we have these unique behavioral patterns. Okay? Patterns, rituals. This is like the most famous one here, the blue-footed booby, which is this bird. And it does this really elaborate behavior mating dance that other birds aren't going to do. And so that helps them choose a mate. The female is looking for a male that does this dance for them, basically. So the other species of birds that don't perform this ritual are not going to mate with the females. Okay, so it's going to, again, identify members of the species. It's going to attract a mate. Okay, and so it's going to keep them reproductively isolated. Some other prezygotic barriers would be mechanical isolation, like they physically don't fit. Okay, so there's a physical issue here that the male and the female do not fit together. Okay, that there is a morphological difference. You can actually see this in these flowers. Okay, so these are two different species of flowers, and so this pollinator right here, this bird, okay, that bird does not pollinate the pink purple flower because of its color, shape, whatever. It doesn't pollinate. So because these two flowers look different, have these physical differences, okay, the bird is not going to transfer the pollen from one flower to another. Okay. There also can be a physical, they mechanically do not fit together to allow the male to release sperm to the female. We also have gametic isolation. So even though the sperm may have gotten to the egg, it may have been released in the same area of the egg, okay, but the sperm is not able to fertilize the egg for some reason. Okay, whether the sperm cannot actually penetrate the egg, so the sperm has to drill through to the inside of the egg for it to be able to fuse with the egg. So it may not be able to penetrate into the egg for it to fuse. Okay? They don't have the right receptors between the egg and the sperm, and so the egg refuses the sperm. It doesn't allow the sperm to penetrate. Or there can be chemical incompatibility, okay, where as the sperm is traveling through the reproductive tract of the female, the sperm may not survive for whatever reason. Maybe the reproductive tract is too long, maybe it's too acidic, but the sperm can't survive in the female reproductive tract. Okay, the common example here are these sea urchins that you see down here on the bottom right. The purple and the red ones are two different species of sea urchins. But sea urchins, while they reproduce sexually, they don't have sex. They release the sperm out into the water, and then those sperm, when they come in contact with the female, they will then fertilize the egg. And so with these sea urchins, when they release their sperm out into the water, the two different species, they don't have the right receptors for the sperm. So they have stayed reproductively isolated, and they never fertilize the egg, therefore we never make the zygote. So in addition to prezygotic barriers, you could have postzygotic barriers. So postzygotic barriers are going to be things where the zygote has formed. So offspring are going to develop, but they either aren't viable, they're not fertile, okay, or they're not producing viable, fertile offspring. Even if they can produce offspring, they're not producing healthy, good offspring. So a postzygotic barrier, the zygote actually formed, but it's not going to work out from there, basically. So examples of that would be this reduced hybrid viability. You see that here with the salamander, right? So the example here would be that this hybrid, this salamander there, it won't survive. It's born, but it won't survive to adolescence, and or it won't survive to adulthood. Therefore, the hybrid is not any good. It's not viable. An example would be sheep and goats as well. Sheep and goats, they produce an embryo, but the embryo doesn't even make it to birth because the chromosome numbers are incompatible. Sheep have 54 and goats have 60. So something is going to impair the hybrid's development. You can also have reduced hybrid fertility. That's what we see in the horse, donkey, mule example. So they produce offspring, and they may produce healthy offspring. A mule is a healthy, vigorous offspring. However, that offspring is sterile. So they're not going to continue to pass on those chromosomes. Remember, that's the whole point, right? That they pass on the chromosomes. And so what happens here is when that mule tries to go through meiosis, it's not going to produce gametes. If you remember, the diploid number of the mule becomes 63. Well, I can't split that in half, and I can't do half a chromosome somewhere. So the mule is unable to produce normal functioning gametes, and so the mule is sterile, even though it's a totally viable, functional organism. Another postzygotic barrier would be hybrid breakdown or elimination. So what this means is that they may be okay at first. So the first generation is fertile and viable, but after they continue to mate and try to reproduce, they get broken down, or they become sterile, they become weaker, they have a lower fitness to the environment, and so they become selected against. Nobody's going to want to reproduce with them because the fitness for the environment is lower. You see this on the far right. Here's the original. Okay, so the first hybrid is fertile, it's viable. Okay, but as it continues to reproduce, you see them getting smaller. They're not as strong. They'll be selected against. So they still remain two separate species, and they remain reproductively isolated. So the rate that speciation and evolution can occur can vary, and it's going to be, some of that is going to be factored into with the ecological conditions. What are the environmental conditions going on at that particular time that could dictate the speed at which change is happening, which could dictate, again, then the speed at which speciation is occurring. So we have kind of two basic ways that we can describe this. We have gradualism, and hopefully you remember these from previous biology, right? We have gradualism, which is that slow, kind of steady change. So evolution is occurring slowly over hundreds of thousands or millions of years, and it's pretty consistent. Okay? Like there's kind of just a slow, steady change from organism to organism. You can see this happening in the butterfly. My morphological change, so the amount of change it looks like is, the amount of change in what it looks like is along the x-axis, where time is along my y. And so I have not tons and tons and tons of change in the x direction, so not tons and change of what it looks like, okay, over time. It's a very slow, gradual change. Where punctuated equilibrium, the evolution is happening very quickly after long periods of no change. Remember, those periods of no change are called stasis. Those long periods of it staying the same. So we, again, morphological changes on the x-axis, time is on the y-axis. So for instance, right here, as time passes, right, I have a lot of time passing in the y direction, but I have no change in x, right? So I have a big change in y, but no change in x. And so I have no physical change even though time is happening. So I will have, however, I have chunks of time where there's very little time happening, right? So no change in y, but a bigger change in x. Okay? And so very rapidly, very quickly, I have a lot of change happening at one time, and then it will stay the same again. It'll go into stasis for a while, and then again, lots of change happening over a period of a very short amount of time. So when evolution is occurring, as these organisms are adapting to their new habitats and potentially becoming their own separate species, we can have what's called adaptive radiation occur. And so when adaptive radiation occurs, I've got my original population left the original group. Okay, so I've got a population that left the original group, and they're going to have a different allelic makeup than the original species. Okay, think founder effect, right? We talked about this. I've got my original population, a small chunk goes off and starts their own population. They don't have the same distribution of alleles that the entire population did. Maybe you took everybody that shows a recessive trait, or nobody that shows the recessive trait. So I get a different allelic makeup than the original population. And so these two groups now are in different environments. So they're going to experience different mutations. And if they're experiencing different mutations and they're also in different environments, I'm going to get different selection pressures. For one population, it may be beneficial to have one trait. For another population in a different environment, it may be beneficial to have a different trait. So they are now experiencing the effects of genetic drift and selection pressure because they have the founder effect that's occurring, and they've got these different selection pressures against them. And so they can eventually, over time, start to develop into a new species. Speciation during this adaptive radiation time can be very, very quick. Okay? Again, think about it like I'm radiating out, right? Here's my center population, right? This ancestral finch that's right here in the picture. And some went to that island, and that island, and that island, and that island. And so on these islands, right, they had different selection pressures. And so different mutations became better than others. We can see the beaks in particular, right? Based on the food supply that was there, it was better to have a certain kind of beak. And so over time, all of these finches have actually become a whole. Each of them is its own separate species. They are reproductively isolated from one another because they radiated out like that and then, again, adapted to their particular environment that was there over time with that population. So again, we'll practice with this in class. We'll write about it. Like always, we're going to be doing a lot of writing about it. Okay? And just practice with a lot of the vocabulary and clearly articulating what's happening in different scenarios about this.